to build a trilobite or a fish or a comb jelly or any of the animals that appeared back then or now, we know we need genetic information. Hey, I'm up here, I'm kind of jealous because all my colleagues get to talk about really cool animals like honeybees and butterflies, and Ray Bolin was doing woodpeckers, and I just don't know anything about these modern animals. The things I know about are these things like trilobites, and this little guy who was discovered in the Cambrian period called Wiwaxia, and primitive fish. And so you may know I wrote a book about the Cambrian explosion, and we just saw a little video partially about the Cambrian explosion. Why is that relevant to our theme today about all creatures great and small? And the question of this session, how to build an animal? Well, the first, not the completely first, but almost the first burst of creativity documented in the fossil record is in a period called the Cambrian explosion. And in the Cambrian, the majority of the animal body plans that have ever existed on Earth came abruptly into existence in the fossil, as documented by the fossil record. Now, a body plan is a unique way of organizing body parts and tissues. We could even say cell types, tissues, and body parts. And when we get to this period of the Cambrian, there are all these new animals that come into the fossil record that have never existed before, and each of them exemplify a unique body plan. And so you have the first arthropods, exemplified by the trilobite. Several other groups of arthropods also emerge in the Cambrian with their hard exoskeletons. You also have the first fish, which exemplify a body plan called the chordates. They have an internal notochord or spinal column. Then you have things like soft tissue creatures like the jellyfish or the comb jellies. They are part of the phylum Ctenophora. And on it goes. You have each of these very distinct ways of organizing body parts and tissues come into the existence in a particular animal form, sometimes more than one example in each body plan, but these new body plans emerge. And so the Cambrian explosion raises the question of this session, which is, how do you build an animal? We get all these animals coming online. How were they built? Now, in my book, Darwin's Doubt, and in this little video, I address two mysteries, and they're kind of related. The first is the mystery of the missing fossils. And if we think of the Cambrian explosion, we're talking about the geologically abrupt appearance of these different animal body plans, these different new forms of animal life. And there are various ways of dating the Cambrian explosion. An accepted date for it is about 10 million years. But it happens that some of the phyla between 13 and 16 phyla within the Cambrian arguably appeared within a 5 to 6 million year window. Now, that's still a lot of time, but it's geologically extremely abrupt when you consider the age of the Earth. And it's also biologically very abrupt when you consider how much evolutionary change we can expect to occur on the basis of ordinary mutational processes. There's a branch of evolutionary theory called population genetics that allows us to calculate how much evolutionary change could take place in a given period of time if we know factors like the population size, the mutation rates, and the generation times, the time from parent to offspring. And so when we do those kinds of calculations, 5 to 10, even 40 to 80 million years is not nearly enough time to account for the origin of all this new biological form by reference to the standard Darwinian processes. Now, I'm going to toot my own horn a little bit because I'm getting I'm not tired of being introduced as having been on Joe Rogan. I mean, it's fun and everything. But I have done other things in my life. But yesterday I got another shout out on Joe Rogan, and somebody just sent me the text, so I thought I'd share it with you. I have a friend who's an evolutionary biologist named Brett Weinstein. He teaches he was at Evergreen College in Washington State. He's fairly prominent. He's on Rogan a lot. And he's a fair-minded evolutionary biologist, and we've had a number of exchanges, conversations at conferences. We enjoy each other's company. He's a good guy. But he's on the other side of the intelligent design, undirected evolution controversy. He believes that some undirected evolutionary process can explain the origin of new animal form. But on Rogan yesterday, he said he said, Stephen Meyer, whom you've had on your show, he referred to me to Rogan, he said, he believes that the evolutionary process, the Darwinian process of mutation and selection, doesn't have the power, by which he meant the creative power, to build all these new forms of animal life. And then he said something extremely telling. He's not wrong, he said. He didn't come right out and say he's right, but he said he's not wrong, okay? Now, Brett has, as I said, he's a fair-minded guy, and he understands a good argument when he hears it. And he says, well, we're going to find what the mechanism is, but we just don't know it. He's also said, you know, I met these ID guys at several conferences. He knows Paul Nelson, for example. He knows Rick Sternberg. He said they're smart and they like science. And he said, and if we don't take seriously the challenges that they're posing to evolutionary theory, we're never going to solve the problems. This was on a previous Rogan episode. So this is just all by reference to, I mentioned the 2016 conference where leading evolutionary biologists have convened to address the need for a new theory. Brett Weinstein is, I don't think he was at that conference, but he's in that wider group of evolutionary biologists who are saying, Houston, we've got a problem, okay? And he's saying we're not going to solve the problem until we acknowledge it. And even if it's these pesky ID guys who are pointing out the problems, we've got to address them. So that's kind of encouraging, and some context of the discussion. In any case, in addition to the first problem, the first mystery associated with the emergence of animal life is the pattern that we see in the fossil record. We see not only this abrupt appearance in the Cambrian period, which contradicts the Darwinian picture of the tree. The tree is a picture of the history of life in the Darwinian scheme of things. So the Darwinian tree is meant to represent all the forms of life today. Those are represented by the branches at the top of the tree, and the trunk or the base of the tree represents the first organism from which all the other branches emerged in a very slow and gradual and completely naturalistic way. But unfortunately, for the Darwinian view of things, that's not what we see in the fossil record. In the Cambrian period alone, we see this abrupt appearance of a couple dozen, at least, animal body plans. And with no, and this is the key when I say abrupt, I mean with no connection to any discernible ancestors in the lower Precambrian strata, the sedimentary layers. And so, whereas theoretically we should see something that confirms the picture on the bottom of the screen, what we see, in fact, is something that looks much more like on the top of the screen. And you can see there's a big disparity there between those two ways of demonstrating or depicting the history of life. Now, Darwin himself was aware of this all the way back to 1859. In The Origin, he writes, to the question why we do not find rich fossiliferous deposits belonging to these assumed earliest periods prior to the Cambrian system, the Cambrian layers, I can give no satisfactory answer. So he acknowledges it as a problem. I talk a lot about what's become of, I call this Darwin's doubt. It wasn't a doubt about whether his theory was true. He was convinced it was, but he doubted that it was an adequate explanation for all classes of evidence, including especially this one. The problem of the Cambrian explosion, though, has become even more acute in the ensuing years. Fossil investigations have not turned up the ancestral precursors that his theory requires, and the period of time associated with the abrupt appearance has gotten shorter and shorter. Most of the major groups appear within 10 million years. A big bunch of them appear, as I mentioned, within an even shorter window of time. And it's not enough time for the mutation selection mechanism to do its alleged magic. And that raises the second mystery that I address in the book Darwin's Doubt, and which is the topic of this session and, in a way, a culminating session for our our theme of all creatures great and small, and that is, how do you build an animal? Or how would the evolutionary process build an animal? Or put it even generically in a theoretically neutral way, what is required to build an animal? Well, the little video I showed for a reason, and that is it highlights one thing we know you need. To build a trilobite, or a fish, or a comb jelly, or any of the animals that appeared back then or now, we know we need genetic information. Because all of these animals are built they have unique organs and tissues and cell types, but each of those unique cell types has distinctive proteins in them. So for example, when you get the first fish discovered in the Cambrian period, which was discovered in the Chengjiang formation, the Maoshishan formation in Xinjiang, China. This was discovered fairly recently, within the last 20 years or so. The first fish came online. Well, what do we know you need to build a fish? Well, fish have muscles. And so you need myosin, an important protein for building muscles. The trilobites, they have hard exoskeletons. There's a whole series of proteins that are needed, one of which is called lysyl oxidase, which links together various other proteins that are needed to make a hard exoskeleton. If you get also a fish or many of the other organisms that had digestive tracts, if you have a feeding tube or a digestive tract, you're going to need digestive enzymes. Enzymes are proteins. So where do proteins come from? Well, if you watched the little video, or were here last year and heard my talk, you know that proteins are built from the information encoded in DNA. That the information in DNA directs the construction of proteins in the cell. So the first thing you need to build a new biological form, a new animal form, is information. You need the information to inform the organism, to build the proteins that keep cells alive. And so in the little video you saw, I reprised the discussion of Watson and Crick and their discovery of the double helix, and more importantly, Crick's sequence hypothesis, where he realized in 1958, 5 years after the elucidation of the structure, that DNA contained information as we saw in the video, in a digital form, a digital or alphabetic form. And that information is necessary because it directs the construction of proteins and protein machines in cells. So one of the big questions that arises when you see these big bursts of biological innovation or creativity in the fossil record is, where did the information come from to build all the new body parts and cell types, and in particular, the proteins that service those cell types that make them functional? Where did the information come from? When you see a quantum or abrupt appearance of biological form, we know now, because we understand the inner workings of the cell and how animals are put together, that entails the need for an abrupt or a big jump in the amount of biological information that's on tap. There's a quantum increase in biological information that's represented by the Cambrian explosion. Now, last year I went into a great deal of technical detail about why it is difficult to explain the origin of new information in DNA, for example, a stretch of DNA for building a protein, why it's difficult to explain the origin of new information from a pre-existing section of DNA. So if you've got DNA A and you need to build DNA B, DNA A builds protein A, you want to build DNA B so that DNA B can build protein B, you've got a problem. How many computer scientists or computer engineers or software people do we have in the audience? Quite a number. We always get a good bunch for this. Let me ask you a rhetorical, hypothetical question. If you have a section of code for building a program that works, what happens if you start randomly changing the zeros and ones? What will result? Will you get a new program or operating system, or will you degrade to the point of destruction the original program that you had already? Okay, it's obvious. I mean, I love a good rhetorical question that causes everyone to go like this. We know the answer. I know, teach. Okay, yeah, it's pretty obvious. Now, in the video I showed there's a reason for that. There's a mathematical reason in all codes and languages. The number of ways to go right is much smaller than the number of ways that there are to go wrong. And so if you start randomly changing things, you're inevitably you're going to fall into the go wrong bucket.
You're going to get a sequence, and you might get lucky with 1 or 2 changes, but the more changes you make, the higher the probability is that you're going to destroy the function you had long before you ever get new function, okay? And so that's the problem I exposited in probably excessive detail last year, okay? It was a new technical talk I was trying out on y'all. But the basic idea is pretty clear. What's the basic idea? That the neo-Darwinian mechanism of random mutation, of natural selection acting on random mutations and variations is insufficient to generate the new information needed to build the proteins that are necessary to service new cell types and in turn new forms of animal life, okay? And if to be clear on why that's the case, think of that computer analogy. Start randomly changing the zeros and ones, or the A's, C's, G's, and T's, you're going to degrade function before you ever find new function. Now, that is not the only problem, though, about how to build an animal. Paul Nelson was talking in the Q&A this morning about the things that the developmental biologists do to these poor, hapless fruit flies. And it is very sad. The biologists have these fancy euphemisms. They call them mutagenesis experiments. It means torture, okay? They radiate the genome. They expose the genome of these fruit flies to chemicals. They expose them to various temperatures to induce mutations in the fruit fly genome. And there are 2 great scientists, Christian Nusslein-Volhard and Eric Wieschaus, who worked on this in the mid-1980s. They did mutagenesis experiments on Drosophila, one of what biologists call model systems. That means organisms that we don't mind torturing. And what they did was they induced a lot of mutations in different sections of the fruit fly genome. That's what Drosophila is, a fruit fly. And we're able to see what the different mutations in different parts of the genome did to the ontogeny of the organism, to the development of the organism. So if they mutated one part, they might find that it ruined a leg. Well, then what did they figure out? Well, that part of the genome is involved in building the leg. If they did something that caused a mutation called antennapedia, well, okay, that has something to do with the antennae. And then do another one that messes up the vision. So by mapping, by correlating the location of the mutation to the phenotypic effect, the effect on the visible organism, they were able to map the genome and to figure out what it did, which parts of the genome were involved in in constructing which parts of the fly. It's a fantastic piece of scientific work, and they got the Nobel Prize, and rightfully so. It was fantastic. But they also discovered something very unexpected, and that is that if they mutated the DNA early in the process of embryological development, just a little repairing back to maybe some high school science, you know how embryos go through cell division. You get a fertilized egg, and then it goes to 2 cells, and then it divides 4, and then to 8, and to 16. It's a geometric expansion in the number of cells, okay? If you mutate the DNA in those early cell divisions, inevitably, the fly died. You would not get to full development. It would shut down. Development would shut down. Now that created a big puzzle for, here's the geometric expansion, the number of cells. Nice little animation. That creates a big challenge for evolutionary biology, evolutionary theory. Why? Well, because if you want to change an animal body plan, if you want to make a large-scale change to the form of an animal, you're going to need necessarily to generate changes in the earliest cells in that process of cell division. Why? Because if you wait till later, you've had differentiation. By the time you get to the full complement of cells, if you get a mutation in those somatic cells, first of all, they're not going to be heritable, but secondly, they're only going to affect a very small region of the organism. So if a mutation is induced in the DNA of a skin cell, it's not going to do much. To make a global change, you've got to change things early. You've got to mutate things early. so that everything downstream is affected and you can get a large-scale alteration in the form of the animal. But what these two scientists found was that when they mutated the organism early on, invariably the whole system crashed. So what's the moral of that story? Well, the kind of mutations you would need to build a new animal body plan, you don't get. What kind of mutations do you need? You need favorable body plan altering or early acting body plan altering mutations. We can induce early acting mutations that alter body plans, but they always alter the body plans in such a way that the organism doesn't survive to full development. Meanwhile, the kind of mutations you don't need, mutations that might occur much later in development, that only affect a small cluster of cells in a localized region, or which are not heritable, now we get those mutations in abundance. But they don't build new forms of animal life. So the kind we need, we don't get. The kind we get, we don't need. It's a great geneticist named MacDonald called this the great Darwinian paradox. So a lot of times when you get in an argument about whether or not mutations are or are not sufficient to build new stuff, don't get trapped into arguing about mutations generically. Talk about where the rubber meets the road, which is the early acting mutations that have the potential for changing body plans. What we know about those is they are always deleterious. There may be some other mutations that are not always deleterious. They might cause small changes that are advantageous in some ways or other, but the ones that would be needed to build a new body plan are not. And that's a big problem. So it's a problem for the Darwinian account of how we build animals, or how the evolutionary process would build animals. Let me give you another related problem. And this, I just love this. Turns out that there are things that, when animals are developed, the development of the organism is orchestrated by something called a developmental gene regulatory network. A developmental gene regulatory network is essentially a relationship between specific genes and the signaling molecules that are produced by those genes. So genes produce gene products. Some of those are proteins. Some of those are RNA molecules, which in turn are signaling molecules that turn on or turn off other parts of the genome. So as the organism is going through that 2, 4, 8, 16, et cetera, developmental process, as more and more cells come online, those cells start to differentiate themselves one from another. Some become bone cells, some become muscle cells, some become some skin cells, some produce hair follicles, some produce internal organs. And each one of those different cell types needs dedicated proteins to service the function that it performs. And so what's going on genetically, or at the level of the genome, is that different parts of the genome have to be activated to produce those specific proteins in those specific cells here to differentiate those muscle cells from those nerve cells over here. And over here, the nerve cells are being activated by something else. So all this is tightly choreographed so that the right cells are being differentiated one from another at the right time, and the right proteins needed to service those cells are being produced at the right time. So a developmental gene regulatory network is in effect, it's in effect a circuit. It's a circuit that's controlling the turning on and turning off of genetic information. And so when the developmental biologists, in particular one named Eric Davidson at Caltech, worked some amazing work in developmental biology, when they mapped these systems of genetic control and mapped the functional relationships between the genes and the gene products and what the gene products were doing in turn to either turn on or turn off or regulate or upregulate or downregulate the genome, and they mapped all that, it came out looking like this. Any engineers in the room? What's this look like? Looks like a circuit, doesn't it? In fact, it's an integrated circuit, but not an integrated circuit that is controlling the flow of electricity. It's an integrated circuit that's controlling the flow of information. And it's also mapping functional relationships between the different parts of this. It's absolutely fascinating. You talk about fearfully and wonderfully made. But here's the rub for evolutionary theory. When Davidson and some of his colleagues experimented with the elements, the core elements of these gene regulatory networks, what they found is that if they perturbed them, if they altered them, if they subjected them to the genes that were involved to mutations, that again, animal development would shut down. There's a principle of engineering that many engineers, especially electrical engineers, will be familiar with. It's called the constraints principle, and it goes like this. The more functionally integrated a system, the more difficult it is to perturb the system without defect to the whole. And what Davidson and Britton and his various colleagues at Caltech found was that principle applies in spades to the gene regulatory networks. You perturb the core elements of that, the whole system shuts down, and you don't get a fully developed animal. Now, let me use my hands as a visual aid here. Imagine we now know, so another answer to our question, what does it take to build an animal? Well, you need proteins, but you also need gene regulatory networks to control the timing and expression and production of those proteins and other gene products. So to build an animal, you have to have a gene regulatory network. Let's call it gene regulatory network A. And you want to build body plan A. All right, so far so good. But what if you want to mutate, change, evolve body plan A into body plan B? Well, that means you're going to have to alter the developmental gene regulatory network. But what's the one thing we know from experimental evidence, from experiments, that developmental gene regulatory networks do not abide? They do not abide that kind of alteration, perturbation, that kind of change. So to get to animal body plan A from gene regulatory network A, so far so good, but to get from one body plan to another, you're going to have to alter these gene regulatory networks. You're going to have to get from one to another, which means you're going to have to perturb it, which means you're going to end up destroying the development of the first animal long before you get to the development of the second animal. Do you hear in that an echo of the same problem we have at the protein level? That there are these functional abysses in between stable structures in what sometimes is called morphospace or functional space. Functional animal in circuit A, functional animal in circuit B, but in between, an abyss, all right? Big problem. And it's a problem, Davidson has said, of blessed memory, he passed away, but he said, contrary to classical evolutionary theory, the processes that drive the small observed changes as species diverge cannot be taken as models for the evolution of body plans, the body plans of animals. These are apples and oranges, these processes. Small-scale variation is not the same thing as large-scale innovation, and the processes that drive that small-scale variation are similarly not the same as the processes that would be required to build a fundamentally new animal form. And he says actually that neo-Darwinism, he's been quoted as saying that neo-Darwinism is a catastrophic error in thinking because it does not address this problem. It turns out that none of the other leading evolutionary models do either. And we'll get to that. So this is a ubiquitous problem across all of evolutionary theory. Nobody has touched this. The theistic evolutionists don't touch this. The punctuated equilibrium people didn't solve this. People doing natural genetic engineering don't solve this. The new synthesis doesn't solve this because the problem is basically you can't touch this. These systems do not abide change or significant change. But what do you need to explain significant change? So whatever the mechanism of change is, it doesn't matter. You can't change these systems appreciably without destroying the original functional animal form. All right, there's one more on this. What is our stop time? We got to go. You know what? We could talk more about this. I'm going to just breeze through this real quickly. It's called ontogenetic information. It turns out that in addition to the information you need to build proteins, that's not enough. Because a protein by itself does not an animal make. So in addition to the genetic information you need to build proteins, there are other forms of information that are needed to organize proteins into what are sometimes called biosynthetic pathways, systems of proteins that do specific jobs, like the vision cascade that allows us to see. And you need to organize, and those biosynthetic pathways often characterize distinct cell types. Those cell types have to be organized into distinctive tissues. The tissues and cell types have to be organized into organs, and the organs and tissues and cell types have to be organized into body plans. And the information to do all that organization at those higher levels is not all contained in the DNA molecule, which means that you could mutate DNA indefinitely till the cows come home without respect to probabilistic limits or anything like that, and you would never build a new animal body plan. Because DNA alone is not sufficient. It's the wrong tool for the job. DNA will, in the best of cases, if you can overcome all those long odds we were talking about earlier, in the best of cases, it will just give you a new protein. But you still got to figure out how to organize the proteins into the cell types, and the cell types into the tissues, and the tissues into the organs, and the organs into the body plans, the house that Jack built. So this is a way of depicting this idea of hierarchical organization of information required to build an animal and an animal body plan. So what do we need to build an animal? Oh, and we can analogize this to, say, a computer network where we've got information for building low-level chips, but other information necessary to place the chips on the right place on the circuit board, additional information that's necessary to place the circuit boards into the whole context of the construction of the computer, and then maybe the computers have to be organized into a network. So we see analogies to this kind of technology in our human high-tech digital world. The hierarchical organization of information is the problem. Point is, none of the leading evolutionary theories, not old-line neo-Darwinism or any of the other theories, address these fundamental three problems I've just been talking about. Now, very quickly, you've probably heard me speak before about the logic of the design inference, or one way of unpacking that. And one way to think of that is that in the historical sciences, when we're trying to figure out what happened in the distant past, we make inferences to the best explanation, where the best explanation cites a cause which is known to produce the effect in question. This was a method that was pioneered by Charles Darwin, drawing on the work of the pioneering geologist Charles Lyell, who said that when we're trying to determine the causes in the remote past, the causes that have caused things, when we're trying to explain events in the remote past, we should be looking for causes that we know are in operation, causes that are known to have the power to produce the effect in question. I just want to ask one question. What is the cause that we know that produces digital information? Well, we know that if DNA is like a software program, it's very suggestive that maybe it was a programmer of some kind, an intelligence. You've heard me unpack this argument before, but notice the same type of logic can be applied to developmental gene regulatory networks. These are functioning circuits. What is the one cause that we know that produces circuitry? Electrical engineers, intelligent agents. And we have good reasons for doubting that the neo-Darwinian mechanism or any other evolutionary model can produce one form of circuitry from another. So the presence of these circuits in all these different forms of animal life is another positive indicator of intelligent design. And we could say that we could apply the same type of logic in looking at the presence of hierarchically organized information. We don't just find information in the DNA, we find a hierarchically organized system of information processing and expression inside cells and animals. And that too, I think, speaks to intelligent design by the same logic, because we know of only one cause that produces information in a hierarchically organized way. So I'll stop there, and we're going to bring up Paul Nelson, who's the real expert on the developmental biology, and we're going to talk a little bit more about this and have a bit of a conversation to close out our session here. So Paul, why don't you come up, and thank you, by the way, for your outstanding talk on butterflies this morning. I think I mentioned it in another session this morning, but this is the 40th anniversary for me of the day I learned about intelligent design at a conference here in Dallas. And thank you. And only very soon afterwards, I met this guy. We go back to 1988, and long conversations for me from a British phone box, pumping it with coins. I found this brilliant guy at the University of Chicago who's thinking about these things the same way I am. So we've been in this for a long time together, and it's a pleasure to have you here today.
He still has most of his hair.
So Paul, I'd like to start just by talking a little bit more about, you're the expert on this. Most of what I've learned on this, about this, I've learned from you or Jonathan Wells. And these
let's start with the early acting body plan mutation thing, because I think that was something that really lit your fire when you were working with some of your mentors at University of Chicago, Lee Van Valen, Bill Wimsatt, this idea of, what is it called, generative entrenchment? Could you explain that?
Well, I actually have an object lesson because I know Steve loves props. So here's my iPhone 15, and down here I have an extinct charger. Okay, who has an iPhone 15 here? Okay, quick quiz, what's the most important component of your phone with respect to everything that it has to do?
The case on the outside, right?
No. I heard charger, right? If you don't keep it charged, you will have a very expensive paperweight, right? Okay, so my wife goes crazy because I don't throw away extinct charging cords. If you look at the end here, it's a lightning connector. This takes a USB-C, right? So this is not going to happen. No matter how hard I try, I am not going to charge this phone with this cord. So generative entrenchment at its basic level is a theory about functional importance. There are more functional relations coming out of the power supply into the phone as a whole than any other component. Even the CPU is downstream of the power supply. So generative entrenchment says what features of living things are so deeply entrenched that they cannot be modified, as Steve put in his talk just now, without crashing the system. Well, we see that in cell biology with things like the genetic code or the ribosome, but we especially see it in developmental systems like animals. And what I realized, even as a college student, when I was following these experiments by Nusslein, Volhard, and others, is they were breaking fruit fly development at different slices of time, and the severity of the consequences of those mutations would map to the time axis. As Steve pointed out beautifully in his talk, those early acting mutations, if they had global effects downstream, the flies didn't like it. You know, right? It makes perfect sense. If you have a handful of decisions early that have all kinds of downstream consequences, it is probabilistically overwhelmingly likely that those random changes early will cascade downstream and disrupt.
Adversely affect something, right?
Adversely.
Yeah, because so many things downstream depend on it. That one or another of those functionally necessary elements is going to be adversely affected.
And my advisor at Chicago, Bill Wimsatt, has this beautiful theory called generative entrenchment, where he says this is a logic feature of any complex system. So think about your car, that laptop, my phone, airplanes, any complex functional system has these relationships.
So I can take your case off of here and I can
Don't do it.
crack it, break it, and it's not going to have much of an effect on the function.
Phone will be fine.
But if I mess with the CPU, you're not going to be able to call Suzanne today.
I'm not going to be able to call Suzanne, exactly. And when I was a graduate student, two of my advisors, Lee Van Valen, who died in 2010, wonderful evolutionary biologist, and Bill Wimsatt, who was my PhD director, who's still alive, worked on this problem. The problem has been known in evolutionary theory since the time of Darwin. And it has resisted explanation. Today, February 2025, it is still an open, unsolved problem in evolutionary theory. And the reason, I would say, is not that they haven't tried hard enough. The reason the problem is unsolved is generative entrenchment relationships are properties of design systems. They are properties of systems where a designer has assembled components in such a way that some of those components are far more important for the global set of functions that that system has to perform than others. And
and those are the ones you can't alter.
Those are the ones that you can't alter,
change.
or if you do alter them, the consequence is so severe that in terms of Darwinian theory, the fitness drops effectively to zero.
That's a very good explanation, Paul. Let's now talk about the second discovery and development I talked about, which is these developmental gene regulatory networks. You had the opportunity to interact at a very important conference in China with Eric Davidson on precisely this question of how would you evolve a new developmental gene regulatory network, and therefore how would you ever get a new form of animal life?
I spent almost 2 weeks with Eric Davidson in southern China in June 1999. He wasn't very happy that I was there. It's a long story, but I'll give you the short.
But that's an interesting thing. Part of this,
Yeah.
he was very anti-neo-Darwinian, but it wasn't because he was a creationist or a
No,
theist of any kind. He
was
didn't like intelligent design. He just knew that the received theory didn't work to explain these things.
So I'll give you one anecdote of 2 weeks worth of interactions. I'm sitting outside the dining hall waiting for lunch one day, and I have an overhead that I made of a gene circuit that his lab had worked out the logic for. The gene was called endo-16, and it's involved in building the gut in the purple sea urchin. He single-handedly, he and his lab, figured out how the purple sea urchin develops. And what the wiring diagram is, as Steve showed, actually that one that he showed you on the slide, that is for the purple sea urchin, for specifying cell types and tissues. So I'm sitting there and I have this overhead, and he's walking up with his girlfriend, and about 10 feet away, he sees the figure, and he knows it's his. And he goes, "What are you going to do with that?" And I said, "Well, I thought I might throw it up on the overhead and ask people, what do you think constructed this very complex regulatory logic?" And he goes, "Well, I can tell you what didn't construct it. Darwinian single-base pure mutations didn't construct it." And
So he said, "You were not wrong."
he said, "Yeah." I really, it's probably not too strong a verb to say that I loved what he did. Every paper he wrote was interesting. He was brutally honest. And he was very tough on his colleagues. So some of you heard earlier today about Jerry Coyne at University of Chicago. Jerry and Eric Davidson were like this, because Davidson is a classical Coyne is a classical neo-Darwinian, Davidson is not. And Davidson said, "Look, textbook theory is bust. It just doesn't work." And he went to his death. Unfortunately, he was a very heavy smoker. He went to his death with this problem unsolved. And I actually have a couple of slides. Do you mind?
Please.
If
You said, well, go ahead.
Yeah, I'll just
click
advance
it.
them. So one of the reasons we were there, here we go. This is a family tree of the animals. And I want you to focus, evolutionary family tree of the animals. I want you to focus on this group right here, the ecdysozoa. These are animals that undergo molting. So that name literally means, ecdysozoa means the molting animals. So we're going to look at two groups, the arthropods, which is where fruit flies are located, that phylum, and the nematodes, which are, I knew this might happen. Those red dots are supposed to be on last common ancestor of ecdysozoa, and then the next node where they all come off. But they got shifted, it doesn't really matter. Let me show you what they look like. Here is our fruit fly, here is our nematode. And these are very, very well-studied model systems, probably better known than any other animals right now in biology. But I want you to look at how they develop. So here is a fruit fly embryo, very, very early in development. It's just been fertilized. And that is one big cell. It's called a syncytium. There are no cell membranes at that top stage. What's happening is the nuclei are dividing, and they are migrating to the periphery of that one big cell. Only when they reach the periphery in the third box do cell membranes form. Okay, so keep that architecture in mind, the one on the left. Now here is how C. elegans, the worm, develops. After fertilization, the very first event is a cell division. all right? And then those two cells divide to 4 daughters. These architectures could not be more different. In one, you have one big cell with nuclei migrating out. In the other, the first event, you have a cell division, yet they were supposed to share a common ancestor. Now, anyone in this room can see the puzzle here. If these two groups, as that family tree showed, shared a common ancestor, one of them is going to have to radically modify its early development. Dramatically. Right? These are not small-scale changes. You're changing the fundamental architecture of this animal. Now, in China, Davidson is trying to solve this problem in the framework of common descent. He is not going to give that up. The animals shared a common ancestor because the alternative was unthinkable. And he unfortunately went to his death with this problem unsolved. And I'll tell you, it will never be solved in an evolutionary framework, because the signal from nature is you cannot perturb these early stages and get a functional animal out of it.
So this is another can't get there from here problem.
You can't get there from here. And even if I weren't a Christian, even if I weren't a theist, if I were an atheist, I would still hold this opinion because it's what the evidence tells us. And when biology comes to a point like this, where basically a philosophy is standing in the way of the advance of the science, and you're trying to solve a problem where the animals themselves are telling you this is not going to happen, that's when you are walking down an alley and your nose is bloody because you keep hitting the brick wall at the end of the alley, right? And the rational thing to do is to turn around and go back out, and to change your background assumptions. And I've been doing this for 40 years. You know, I used to be skinny and have more hair. And this guy eats like a horse, and he's still skinny, and I don't know how he does it. Anyway, so you can see I'm passionate about this because, you know, nature is reliable. She is I like to personify her as a very beautiful, very stubborn woman. And this is probably not a PC analogy, but what the heck, you know. And you think you've got her locked up in your basement, and she picks the lock and goes on her way. Nature talks back to science, and if science doesn't listen, nature is not going to change the story she tells. And until we listen to her, we're going to be stuck.
The same dead ends we've had for 40 years.
Same dead ends.
Paul, before you go, let me ask you, are there any other examples of this can't get there from here? You were telling me something on the phone the other day about the ribosomes.
Okay, so this is not as important as other aspects of your cell biology, but this defines life on Earth. Viruses, by definition, do not have ribosomes. Ribosomes are the big factories in your cells that take your genetic information in DNA and RNA and turn it into protein. Incredibly complicated, interesting machines. I've really fallen in love with them. So when I was a student, there was the bacterial, note that definite article, the bacterial ribosome and the eukaryotic ribosome, which you see on this slide. On the left is the bacterial, on the right is the eukaryotic, and they have two major components called the large and small subunits. The small subunit is the decoding part. That's where the messenger RNA strand comes through, and the decoding unit recognizes the individual codons, which will then be assigned to amino acids. The large subunit is where the bonds are actually formed, where transfer RNA comes in, carrying amino acids, and you string together those amino acids and you make a protein, all right? Essential for all life as we know it. If there are viruses on Mars, there will only be there if there are ribosomes on Mars. So what a virus does is SARS-CoV-2 did this with a lot of us, right? It comes in and hijacks your ribosomes because it needs them to make its proteins. Okay, so I'm a student, the bacterial, the eukaryotic. Well, science moves ahead, right? It doesn't stop. Today, in February 2025, there are remarkable differences among ribosomes all throughout life, and they are dramatically different. not just a few little things different, dramatically different.
Is that in structure, Paul, in the parts?
It's in structure, it's in function, it's the same machine in a generic or abstract sense.
Yeah, it's performing
namely,
the same basic function of,
same basic
generating
function.
proteins.
But just like with my iPhone 15 and that extinct chord that I can't use, one of the features of the logic of complex systems is as you move, and Steve alluded to this in his presentation, as you move through functional space, you can reach a point where you create a discontinuity, right? And that discontinuity cannot be bridged by any gradual path because you have to jump it via design. So in the book that I'm working on right now, the big ending discussion is on this. And I want you to buy the book. So I'm not going to give away all the best jokes, right? But it turns out that ribosomes also do not like to be tweaked. They really don't like it. These are very complex factories. You can make some small-scale changes, but if you try to modify them in the ways that seem to be required to give us the diversity of ribosomes we observe today, the ribosome talks back to you and says, you know what? You couldn't change those proteins because I needed them for my functional role. So if you pursue that logic through to its conclusion, what you see is there may be discontinuities in nature that are built into the very foundation of living things. And we can discover that by trying to perturb them.
Here, let's just discuss
So,
this a little more. This is
yeah.
fascinating because there's an echo again of this same problem. We saw it with getting from protein to protein. We saw it from getting from DGRN to GGRN. And now we're seeing it with a molecular machine, a crucial molecular machine. So you're saying that within the taxonomic landscape of living organisms, it's not just two forms of ribosomes anymore. There's multiple types of ribosomes from the structural standpoint, from the way they're put together, but their functional logic is the same. They're producing the same kind of outcome. They're taking genetic information, they're producing proteins, but the different ribosomes in different types of organisms are built differently. And second point, if you alter ribosome A to try to make it into ribosome B, you destroy ribosome A before you get to ribosome B.
Exactly.
So it's the exact same problem as with the DGRNs.
Exact same problem. And one of the advantages that we have today is DNA sequencing technology and other biomedical technologies have enabled us to visualize these differences in a way that wasn't the case even when I was a graduate student. There's a lab at Yale that does a lot of this work, and I remember where I was sitting in a Starbucks when I read one of their most recent papers, and I was so excited. It was like goosebumps, and I grabbed my phone and I start texting all my friends who work on related problems, and I say, you got to go get this paper. Because what they describe is the diversity present in ribosomes now, and it won't go away. Because each time we look at a new group, we find more diversity.
But what is that signal telling us? Because we've been told that all life arose through a process of continuous biological change, such that the history of life can best be depicted as a great branching tree, a single tree, what in biology is called a monophyletic view of the history of life. But if there are, even at the molecular level, if there are systems that can't be altered without being destroyed, and if there are animal forms that can't be altered without being destroyed, or circuitry that can't be altered without being destroyed, that suggests a discontinuity that means that you can't have a continuous morphing and changing and hope to maintain the function that's necessary for natural selection to preserve something and pass it on.
I couldn't say it any better than that. If there are discontinuities in nature of this sort, that's what biology needs to come to grips with.
But what's that do with our picture of the history of life then?
It radically shifts it. In the original sense of radical at the root. It shifts it at the root. And one thing that's happened that I never would have expected as a student is very senior, I'm talking National Academy of Science level evolutionary theorists, for reasons totally unrelated to intelligent design, are beginning to say Darwin's tree never existed. And what's happening is that the tree of life is coming apart at its base. And you know what happens if you cut a tree at its base, right? The fracturing is impossible to stop. It goes up into the trunk and into the branches and so forth. So you guys, the people in this room, are living at a period in scientific history within, I would say, the next decade, you're going to see things that will shock you.
It's a radical paradigm shift.
It's a radical paradigm shift. And
So it's not just a problem. The problem is not just that we don't have a mechanism that can account for getting from ribosome A to ribosome B, or from developmental gene regulatory network A to DGRN-B. It's not just that there's not a mechanism of transition. It's that because there's no plausible way of getting from A to B, what we're looking at is a picture of life. Life looks discontinuous, and therefore the history of life cannot be depicted as a continuous branching tree, but rather you've got discontinuity, which is what we see in the fossil record as well.
Yeah.
And that seems to suggest something like multiple separate origins.
It's the reality of discontinuity, and physics came to grips with this in the last century with the quantum revolution. And I think the problem for many biologists is the alternative to the continuous tree of Darwin is terrifying. It is terrifying to them. It looks like creation.
It looks like multiple.
It looks like multiple independent, instead of a single tree, a forest of life. And I think we need to have.
Well, that's why we've come up with our own euphemism. They call it monophily. We don't say multiple separate creation events. We say polyphily. Polyphily, actually coined by Ernst Haeckel of all people. It's a different picture of the history of life.
It's a different picture of
But
the
it's one that
history of
matches the fossil record, and it matches the discontinuity we see when we analyze these complex systems.
Yeah. So you're in for an exciting ride, and I wish I were younger because I want to be there too. But I now get senior discounts, right? So, and I'm happy to have them, right? So,
Guys in the pool 3 times a week. Keep swimming, Paul. We need you for a lot longer. Thank you for an expert amplification of the points I'm going to talk.